All right, hi guys. It is chapter 9 time. We're moving into muscle tissue, which I'm really excited about because I think muscle tissue is kind of cool. I am on limited time though. I'm trying to get this lecture in the next 25 minutes. I don't think it's going to happen. I might have to pause it and break this PowerPoint up into 2 videos. Sorry to do that to you. I try not to do that, but I also don't want to rush through it too much because it's really important stuff. Chapter 9 covers muscle tissue. So it's obviously one of our 4 primary tissue types, specialized for contraction. There's 3 types as we reviewed back in chapter 4, I think it was. Skeletal muscle, cardiac muscle, and smooth muscle. This chapter, for the most part, is going to focus on skeletal muscle, but we'll review all of them just in case. Skeletal muscle is voluntary, right? This is our bone moving muscles. Cardiac muscle is involuntary, found only in the heart, propels the blood through the body. Smooth muscle is another involuntary muscle tissue. It's found in the walls of all hollow organs like your stomach and intestines, moves fluids and solids through the digestive tract. It also helps to regulate the diameter of our arteries. So let's talk about the functions of skeletal muscle tissue. It's not just there for movement. That's our big obvious answer, right? Movement. But it's also there to maintain our body position. As I'm sitting here in this chair right now, the muscles, the extensor muscles in my back are working to hold my back up. Otherwise, I'd be slumping over like this, which sometimes I do, but try to keep up. It's also there to support soft tissues. Think about the function of your abdominal muscles. They're there to hold your abdominal organs in. They also guard entrances and exits, right? Our mouth, our eyes, all surrounded by muscles. Rectum, surrounded by a muscle, right? All of our body entrances are guarded by muscles. They also help to maintain our body temperature. When you're cold, what do? You shiver. You're contracting your muscles to produce body heat. The friction from your muscles contracting produces body heat. Also, well, we'll get into all of that when we get into temperature regulation next semester. There's a whole lesson associated with that. Our skeletal muscles also provide nutrient reserves. Although it's not the preferred method of nutrients, our body will break down muscle for energy because our muscles are made of protein. The contractile proteins in our muscle fibers can be used in times of starvation for energy so that we don't die. There's the functions summarized for you. You can pause the video here, go back and answer these 3 questions. Let's talk about the structure of skeletal muscle. The skeletal muscle has several parts to it. So you have the skeletal muscle fibers, which have the contracting piece to them. Then there's connective tissues, there's blood vessels, and there's nerves. So all of these things have a role. Let's talk about the connective tissues first. So the skeletal muscle is basically a bundle of fibers. Remember, fiber is a muscle cell. Basically the same thing, right? Or it is the same thing. So they're wrapped in several layers of connective tissue. The outermost layer is called the epimysium, or epimysium, I sometimes say. The epimysium is, think about chicken, right? You get chicken from the grocery store, and sometimes there's that clear layer on top of the chicken breast, sometimes like attached to the fatty parts they have to cut off. That's the epimysium. It is a connective tissue layer that covers the entire muscle. So if I were to chop off my skin, pull back the skin, pull back the fat, and show you my muscle, you would see the epimysium. The epimysium surrounds the entire muscle. It basically separates the muscle from all the other surrounding tissues. The perimysium, or perimysium, is going to surround the bundles of muscle fibers. So inside of my muscle group, there are bundles of muscle fibers called fascicles. You know this. Again, chicken reference. You cook your chicken and you cut it. And what do you notice about the chicken? It can be stringy. Each one of those strings is a muscle fascicle. Those fascicles are surrounded by a perimysium. Same thing in all of your muscle groups, all of your skeletal muscle groups. Your skeletal muscle group itself is surrounded by the epimysium. Then you have the muscle fibers in bundles surrounded by the perimysium. Again, collagen, elastic fibers, blood vessels, and nerves all surrounding those fascicles. The endomysium is surrounding every individual muscle fiber. So it's another connective tissue layer. It loosely interconnects adjacent muscle fibers. It contains capillaries, myosatellite cells, which are the stem cells for muscle tissue, and axons of neurons to control the muscle fibers. So those collagen fibers extend beyond the muscle the epimysium continues beyond the muscle and forms a tendon. A tendon is just attaching the muscle to the bone. A sheet of connective tissue fibers associated with the muscle is called an aponeurosis. So you have aponeuroses attaching muscles to a broader area, like along your back, along your abdomen, along your hips. Here you see the overall picture here of this guy who has much better muscles than I do. His whole muscle group here surrounded by the epimysium. The epimysium fuses at the ends to form the tendon. Inside you see the fascicles. Here you can see a better picture. Here are all the fascicles. The fascicles are surrounded by the perimysium. Inside the fascicles are bundles of muscle fibers. Each muscle fiber is surrounded by an endomysium. And then inside each individual muscle fiber, whoa, we're getting even smaller now. Inside each individual muscle fiber are myofibrils. Myofibrils are little filaments inside of our muscle fibers. So let's talk about how skeletal muscles develop. They develop from something called a myoblast. A myoblast will basically fuse together to make a very large cell with several nuclei, and the myosatellite cells kind of exist as remnants of those stem cells. They can aid in muscle repair. They can't really make new muscle tissue though. There you see the overall formation, myoblast. Myoblasts fuse together to make a very large muscle fiber and a myosatellite cell. So mature muscle cells are very large. They can be very long. They can be very have a large diameter. And skeletal muscles have many, many, many nuclei, hundreds of nuclei in some cases. The plasma membrane of a skeletal muscle is called a sarcolemma. That prefix sarco is going to be something that we see throughout this entire unit. Sarco is just referring to muscle. Sarco and myo are really the two prefixes that deal with muscles. So sarcolemma is the specialized plasma membrane of our muscle cells. And there we go. Other terms referring to muscle fibers, as I said, muscle fibers and muscle cells are the same thing. The cell membrane is the sarcolemma. The cytoplasm is the sarcoplasm. And the endoplasmic reticulum is the sarcoplasmic reticulum, or SR. So there's those specialized terms related to muscle fibers. Here's our muscle fiber cell. There we go. You see the several nuclei. The striations we're going to talk about. Inside, we have all those myofibrils. Think they're like little sticks inside. Pause the video here. Go back and answer those questions. Let's talk about muscle fibers and myofibrils. So the myofibrils are as long as the muscle fiber. There's hundreds and even thousands in one individual muscle cell, and they're banded, and that gives the muscle its striated or striped appearance. The myofibril, here's more words, myofibril is made of myofilaments. There are two types of myofilaments. There's thick filaments and thin filaments. Thick filaments are made of myosin. Thin filaments are made of actin. So here you see what I'm talking about. If I pull out these little sticks that are inside my muscle cell, I have a myofibril, and my myofibril is made of, here's a better picture, actin, which is the thin stuff. It's this little red network, or thick filaments, which is this bluish purplish. On this picture, you can also see the specialized sarcolemma. Notice the sarcolemma has holes in it. Those holes go down into the muscle cell. We're going to talk about that. This blue stuff is the sarcoplasmic reticulum, or SR. Well, that's pretty much all I need to show you on this picture. All right, so let's talk about a sarcomere. The sarcomere is the functional unit of the muscle fiber. So let me just go back quickly and show you. This whole structure here, where our thick and thin filaments are overlapping, that is a sarcomere. So these sarcomeres are lined end to end to end along the whole length of our muscle fiber. So it has a zone of overlap, which is where the thick and thin filaments are hovering near each other. The Z lines are the boundaries. This is where the thin filaments are anchored, and it's also the boundary of the sarcomere. The M line is where the thick filaments are anchored, and that is the midpoint of our sarcomere. There's also an A band. The A band is the dark area. The I band is the light area. And the H band is the area around the M line. It does not include the thin filaments. It just includes the thick filaments. So here you see the whole thing. Z line, Z is like zigzag, Z line to Z line. Thin filaments are radiating out from both sides of the Z line. Remember, these are staggered end to end to end to end to end. Our M line is in the middle. That's where our myosin is connected, or our thick filaments, these purple lines. We have the zone of overlap where our thick and thin filaments are overlapping. The I band only contains thin filaments or light. The A band contains all of the area where the dark, thick bands are, and the H band is just the thick filaments in the very middle. So let's talk about this sarcolemma in a little bit more detail, and we'll start putting the whole thing together. So the sarcolemma is what separates the sarcoplasm from the interstitial fluid, right? It's our specialized cell membrane. It's selectively permeable, just like any other cell membrane. It contributes to an uneven distribution of positive and negative charges on either side. We'll get into that physiology in a little bit. So changes in that membrane permeability can have a drastic change on the electric charge distribution. You can reverse that charge, or when you change the charge on the inside of the cell, or when you shift the ions around, that charge distribution can then spread throughout the entire cell. Whoa, it's getting intense. Before it gets too intense, let's talk about something called a T-tubule. We were just talking about the sarcolemma. Now we have something called a T-tubule. T-tubule is continuous with the sarcolemma. Sarcolemma is the outer cell membrane. Remember back here, here. I showed you the sarcolemma and how there are these little invaginations, or little kind of spots where the sarcolemma folds in. That's where the T-tubules are. So T-tubules are continuous. They go into the muscle fiber, and they extend into and tightly bind to the sarcoplasmic reticulum. Let me go back and show you. Sorry, I keep flipping here for you. Here's our sarcolemma. T-tubules coming in, and this blue stuff is our sarcoplasmic reticulum that's butting up against the T-tubule. T-tubules here. Can I do it? T-tubule is my little marker, and the sarcoplasmic reticulum are my hands, just like that. Form a little group of three. So sarcoplasmic reticulum is very similar to the endoplasmic reticulum, except it has a special thing inside of it. We'll talk about it. It's not on this slide. It might be on the next one. But it's basically a tubular network, and it surrounds the myofibrils, and it enlarges on either side of the T-tubule to form something called a terminal cisterna. So the pair of the terminal cisterna and the T-tubule form a triad. So marker is my T-tubule, my hands dipping down, and my arms are the sarcoplasmic reticulum. This thick area where my hands are the terminal cisterna. So two terminal cisterna and a T-tubule make up a triad. All right, there it is. That makes more sense than my hands and a marker, right? Sarcolemma invaginating in to form the T-tubule. Here's our SR, enlarged on either side. This whole thing is a triad. Terminal cisterna are the enlarged ends. Whole thing's a triad. So let's talk about what this SR does. Remember I said it's slightly different? Well, that's because the SR contains lots and lots of calcium. And its job is to pump calcium from the cytosol into the sarcoplasmic reticulum. So the sarcoplasmic reticulum holds as much calcium as it possibly can. The beginning of the muscle contraction starts when there's a change in membrane potential, ion distribution, at the T-tubule, and that triggers the release of calcium from the sarcoplasmic reticulum into the cytosol of our muscle cell. So here you see just a general review. Sarcoplasmic reticulum, SR, right? It has lots of calcium in it, and our cytosol is constantly pushing calcium into our SR. You can pause the video here, go back and answer these three questions. Let's go back to the structure of our sarcomere and talk about these thin filaments. So we're trying to paint a picture, right? We kind of got this idea here, we got this idea here, we got this idea here. We're going to start putting it all together. So our thin filaments are attached to the Z lines, right? This is basically the ends of our sarcomere. Our thin filaments are mostly actin, but there's different types of actin. There's G-actin or globular actin, and then there's F-actin, which is filamentous actin. So the F-actin is held together with nebulin. There's a picture in a few slides. Also on our thin filaments, we have regulatory proteins, tropomyosin and troponin. Tropomyosin covers parts of the G-actin. The G-actin have active sites on them. An active site is where something's going to bind. I'll give you a hint, it's the myosin. So these G-actin molecules have a special attachment point for the myosin, but tropomyosin is blocking that site. Tropomyosin is attached to troponin. Troponin is made of 3 subunits. Troponin binds to tropomyosin, so it is stuck on the outside of the tropomyosin, but also binds to the G-actin, and it also has an opening for calcium. So the troponin has three points of attachment. It attaches to the G-actin, it attaches to the tropomyosin, and it has the potential to hook up with calcium if calcium is present. So here you see a little bit of what I mean. Here is my filamentous actin, kind of back there. Our globular actin, which are these little globs. The nebulin, which provides like a backbone. The troponin are these little brown structures here. The tropomyosin is the rope, the twisted rope on the outside. So the active sites on the tropomyosin are blocked by the troponin. The troponin also connects with the globular actin, and the troponin, and the troponin also has an active site on it, or an attachment site for calcium. Myosin is much more simple. Myosin looks like a golf club. Myosin has a tail and a head. The head pivots. Myosin is held in place by a small protein called titin. So, here's a much better picture for you. The nebulin is the backbone. F-actin is on the sides, and our troponin is our tropomyosin is this blue rope, as I said. The troponin are these little brown knobs, and the globular actin, etc. So let's talk about the thick filaments. As I said, they have a core of titin. That titin helps to anchor and connect them to the Z lines. They are elastic and recoil, and they're much thicker than the actin. So the myosin molecule has a tail, which faces the M line, and then the head, which has 2 globular subunits. This head is going to connect with the tropomyosin molecule as soon as the troponin is moved out of the way. So here you see, again, a zoomed in picture of what it looks like. So it's called the sliding filament theory. When the muscles contract, the thin filaments slide past the thick filaments. And otherwise, the H bands and I bands get smaller, the zones of overlap get larger, the Z lines move closer to one another. The width of the A band stays the same because the A band is just where the dark myosin is, the thick filaments. The sliding occurs in all the sarcomeres along the entire myofibril. So if one is just moving this much, if there's hundreds of them, they can move that much. The filaments don't change length. It's just that those Z lines that are getting closer together. The actin and the myosin don't change length. They don't change shape. It's just the sarcomere. So you can pause the video here, go back and answer the questions. Let's talk about excitable plasma membranes. Ooh, sounds awesome, right? Well, all cells in the body are polarized, believe it or not. This means that positive and negative charges are unequally distributed on either side of the plasma membrane. So inside the cell is slightly negative compared to the outside of the cell. An unequal distribution represents something called a membrane potential. It's measured in millivolts, and every cell has its own special membrane potential. Neurons and skeletal muscles have a resting membrane potential around -70 millivolts. So take a look here. We have our plasma membrane. We have the inside part of our cell, the outside part of our cell. If you look and count up all the positives, it's much more positive than the inside, which means our cell is negative. resting potential of neurons and skeletal muscles is around -70. So why is that? Well, our three main ions are in charge of this. Sodium, which is in excess in the extracellular fluid. Potassium, which is in excess in the intracellular fluid. Chloride is with sodium on the outside of the cell also. It doesn't really affect too much the distribution because it doesn't really follow into or out of the membrane. There's also proteins that are inside the cytosol that can't cross the plasma membrane either, so they don't really contribute. So our two players are sodium and potassium. So there are leak channels. Leak channels slowly allow the ions to flow down their concentration gradient. So if there's lots of sodium outside the cell, like there naturally is, these sodium leak channels are constantly going to be leaking sodium into the cell, and vice versa. Potassium's in high concentration inside the cell, so these leak channels are constantly going to be leaking potassium out of the cell into the extracellular fluid. Enter the sodium-potassium pump. What does that do? It constantly works against the concentration gradient. It's working to send that sodium back out of the cell, working to send the potassium back into the cell. Here you see the magic happening. We have these leak channels, sodium, potassium's leaking out, sodium's leaking in, but the cell doesn't want it like that. The cell wants that sodium out and the potassium in. So the sodium-potassium pump does that. There can be changes in the membrane potential. They can be voltage-gated channels. So in other words, when a signal comes from another cell, which causes some type of depolarization, some type of shift in ions, that can travel down the entire cell. It can change the entire cell if just one little part has been changed. These voltage-gated channels can detect those electrical charges, and that's what we call an action potential. It's basically a sequence of permeability changes in the cell membrane. So there are several steps in an action potential. The first starts with sodium. There's an increase in sodium ion permeability. In other words, sodium rushes into the cell at the beginning of an action potential. Because sodium's positive and our cell is -70 at resting, this is going to cause the cell to become more positive. Generally around -55 is when the cell says, whoa, we have an action potential going on here. So once the cell reaches what we call threshold at -55, then an action potential will be sent. And that's what causes all of the voltage-gated channels to open, and a huge rush of sodium comes into the cell. This is called depolarization. Our cell is going from -55, -40, -30, -20, 0, 10, 20. It's becoming more positive now. It's depolarizing. When the membrane potential reaches +30, our sodium channels close. Our potassium channels open. And guess what? Potassium is now going to change positions. It's called repolarization. So in repolarization, the voltage-gated channels begin to close, and the sodium-potassium pump will start pumping the sodium and potassium back into their proper positions. So let's take a look at it here on this graph. You have -70. You're resting, right? Something happens, so some sodium channels open, boom, we get threshold. Oh my goodness, now sodium channels open, sodium rushes into the cell, we're depolarizing, we're becoming more and more positive. We hit +30, our sodium channels close, and our potassium channels open. So when the potassium channels open, the cell repolarizes because now all of the potassium is leaving the cell, and our cell's going back down to negative. Now slowly our potassium channels begin closing, and there's kind of this little refractory period, like a hyperpolarization that takes place, and we let a little bit too much potassium out, but that's okay because our sodium-potassium pump gets everything back to normal. So this whole thing can be propagated. We have action potentials that are propagated down an excitable plasma membrane. The neurons and skeletal muscles have what's called an electrically excitable membrane, which means a depolarization and repolarization can produce an action potential. This action potential is going to propagate or continue across the plasma membrane. Happens in less than 2 milliseconds, very, very quickly, and only in one direction. So it can't go from the muscle cell to the nerve cell, it only goes from the nerve cell to the muscle cell. This is allowing for super rapid communication between the nervous system and the muscle system. only goes in one direction. You can pause the video here, go back and answer the questions. Moving right along to the neuromuscular junction. This is the connection between the motor neuron and the skeletal muscle fiber. So the skeletal muscle fiber only contracts when the neuromuscular junction tells it to. There's only one neuromuscular junction per muscle cell, although one motor neuron can innervate several muscle fibers. It's called a motor end plate or a functional group. You can watch this video when you go to mastering. They're not included in the video here. Do I have the sequence? I don't have the sequence. Here you see, and again, I don't know why this picture is so small, but here you have your motor neuron innervating down to our muscle fiber, and you can see all these little axons coming off. This is zoomed into the axon terminal, and here you see its connection with that sarcoplasm, or sarcolemma rather, the outer cell membrane, and we'll look more closely at what's happening there. So these are the major components of our neuromuscular junction. You have the synaptic terminal of the motor neuron. This is having special vesicles. The vesicles have acetylcholine or ACH in them. That's the special neurotransmitter for muscle cells. The motor end plate of the muscle fiber has several little creases which contain acetylcholinesterase, ACHE. ACHE is the enzyme that breaks down ACH after it's been released. The synaptic cleft is just this space between the nerve cell and the muscle fiber that also contains ACHE. So here again, we're just adding on to our picture, our motor neuron with all of its axons. Here's our motor end plate. Here's the axon terminal. There's little vesicles in there. You can see them here. These vesicles contain ACH. Our synaptic cleft contains ACHE. The junctional folds are within the muscle cell itself. So what goes on at a neuromuscular junction? First, there's an electrical impulse, an action potential. That action potential comes down the motor neuron or rises the synaptic terminal. What that does is it changes the membrane permeability of the neuron, which is going to allow those vesicles that have acetylcholine to be released. It's exocytosis. So exocytosis of acetylcholine into the synaptic cleft. So ACH is now going to go through the synaptic cleft and bind to the receptors on the muscle cell, on the motor end plate. So that binding of acetylcholine is going to change the permeability of the cell membrane of the sarcolemma, and now the sodium channels open, and sodium now rushes in to the sarcoplasm. That rush of sodium into the sarcoplasm generates an action potential or charge distribution on the sarcolemma. The ACH diffuses away and it's broken down by acetylcholinesterase, and the ACH receptors can then be reactivated. So this is this whole process happening at the neuromuscular junction. We have our axon. Here it is interacting with our motor end plate on the muscle. Our vesicles can be stimulated by an action potential. This plasma membrane changes in charge, and our vesicles will release acetylcholine into the synaptic cleft, bind to receptors, increase in the permeability of sodium. So sodium now rushes into the cell. ACH is gobbled up by ACHE. And that action potential propagates down our muscle cell in one direction. Just goes down that muscle cell. So this whole process begins something called excitation contraction coupling. This is where an action potential that's generated at a motor end plate moves across the sarcolemma. The immediate effect is an action potential. An action potential is an electrical effect. It's very brief because ACH has been removed. Remember, it's gobbled up by ACHE, so it only happens at that one spot. Action potentials will now move down the T-tubules to the terminal cisterna of the sarcoplasmic reticulum. Once that action potential gets down into the T-tubules, the sarcoplasmic reticulum releases calcium. And as soon as that calcium is released, now your contraction can happen. So in other words, as soon as that action potential gets to the muscle cell, contraction is going to occur because of this T-tubule and the sarcoplasmic reticulum. It's called excitation contraction coupling. It's excited, so it's going to contract. Again, this video, you can watch it on mastering. Here you see the sarcolemma, that action potential coming down to the T-tubules, interacting with the sarcoplasmic reticulum, and the sarcoplasmic reticulum is going to release calcium. Here are the questions you can watch. This is the end of the video. And I have to go pick my daughter up from preschool. I didn't think I was going to be able to finish this video, but I did. I hope I didn't go too fast. If I did, that's kind of the joy of having a recording. You can go back and rewatch and re-listen. Have an awesome day, and I'll make another video with the second piece of this chapter. See ya.